Good afternoon, everyone. Welcome to the Clinical Trials Methodology course webinar series. Today's webinar is on study sections in clinical trials. Before I introduce our presenters, I'd like to remind you that if you have a question, please use the chat box to the right of your screen, or you can simply unmute yourself. Also, at the end of the webinar today, please take a moment to evaluate the webinar, and I will provide the link at the end of the presentation. Now I'd like to introduce our presenters today, Dr. Jaideep Kapur and David Wright. Dr. Kapur is the Eugene Meyer III Professor of Neuroscience, Professor of Neurology at the University of Virginia. He is the Director of the University of Virginia Brain Institute. Dr. Kapur has a long-standing interest in understanding the neurobiological mechanisms underlying prolonged status epilepticus. His laboratory studies mechanisms of rapid plasticity of synapses and neuronal circuits during status epilepticus. He is a co-leader of the established status epilepticus treatment trial, an NIH-funded multi-center clinical trial to determine the best second-line treatment of benzodiazepine refractory status epilepticus. Another area of research is regulation of seizures by hormonal fluctuations. He served as the president of the American Epilepsy Society in 2010. He has received the 2013 Epilepsy Research Recognition Award for Basic Science conferred by the American Epilepsy Society. Dr. David Wright is the interim chair and professor of emergency medicine and director of emergency neurosciences at Emory University School of Medicine. Dr. Wright is actively involved in both the preclinical and clinical assessment of traumatic brain injury and stroke. He was a principal investigator of the NIH-funded multi-center phase 3 clinical trial PROTECT, progesterone for TBI experimental clinical treatment. He is the principal investigator of the Emory Hub for the Neurological Emergencies Treatment Trials Network and for the New Siren Network. He is also the co-PI of the Emory Hub for NIH-funded NINDS Stroke Network. He participates in numerous concussion research and technology development endeavors. He has served on multiple NINDS grant review panels for over the past 15 years. Thank you, gentlemen, and go ahead.
Hi, this is Jaideep Kapur. So I'll be talking first, and David will be talking second. And what I want to talk about today is grant writing, and then David will talk about grant review. So first, my disclosure, I have no commercial or financial interest relationships or other conflicts of interest, but I think more significant disclosure is that I currently serve on NIH study section, and I've served on NIH study sections before for several years. This is my second tour of duty on NIH study sections. And what I want to talk about is preparing your grant application with special emphasis on grant applications that are designed to conduct clinical trials. So, and I've divided this presentation according to the way in which your grant will be reviewed so that it runs parallel to the way David will speak about grant review. The first criterion for review is significance. And in explaining the significance of your proposal, it's important that you explain what would happen if you successfully implement that proposal. And you begin that process by defining the problem, and define the problem very clearly in a few sentences, no more than 3 or 4, and then show the impact of solving it. So some of my good grant writing colleagues even begin by creating a subsection where they say problem, and then they go ahead and demonstrate how they're going to solve it. So common approaches in defining the problem is to define a disease by its incidence, economic impact. That's great if you're doing stroke research because stroke is so common, or Alzheimer's disease, which are sort of common disorders. However, when you are dealing with a rare disorder, you have to explain how knowledge gained by studying that rare disorder will lead to much greater understanding of a general principle of clinical management, diagnostics, or treatment. And it's done skillfully. If you've not done it, work with a senior colleague who can help you see the broader impact of your research beyond the narrow question that you will address in a grant. Again, define the impact of your success, because in assessing overall impact of the grant and significance of the grant, NIH asks us to consider what would happen if this project was successfully completed. Second area of where your grant will get scored is innovation. And in clinical research and clinical trials, people often struggle with this area because they think innovation means a new mouse, or a new way to develop a mouse, or new technology to advance the field. But your innovation can be technological, methodological, or even conceptual. A conceptual advance is also innovative. So if you're going to use a newly developed technology, or you're first adopting a technology, that's a technological innovation. Other way of innovating is if you have a novel clinical trial design, or you're using a novel outcome for your clinical trial, that's innovative. Finally, if you're challenging an existing paradigm, if successful completion of your study challenges an existing paradigm of thinking about a disease, whether it is diagnosis, treatment, or prognostication, then say that this successfully completing this will change the way we think about this disease. So second, and the most important part of your grant, where most of the grants suffer greater scrutiny and criticism, is approach. So under the approach section, there are several sections, and this is where your reviewers will focus quite a bit on. And the first section of approach is to show your preliminary data. It's your opportunity to demonstrate that you're capable of performing this study. A review panel consists of very skeptical people who are very skeptical of your capability to finish a proposed plan. This is because they've seen many, many good ideas not come to fruition because they were not feasible. So they look at feasibility. And in clinical research, there are 3 R's that stand in the way of conducting a clinical study. These are recruitment, retention, and regulation. So make sure that you show that you can recruit sufficient number of subjects, which is going to come into your statistical design later. Make sure that you have a plan for retaining your subjects. If you can't retain your subjects, how will you deal with it? You have to inflate your sample size for retention. Finally, regulation is important. If you're using a new drug or a drug which for an indication that is not FDA approved, then you'll need an IND. If you're using a new instrument, you may need an IDE, and you'll need IRB approval. And so making sure that you are aware of those issues and have handled them appropriately prior to submission are important. Again, feasibility of an assay is important. Very recently, I was thinking of doing a study on the impact of progesterone on seizure withdrawal, and I said, oh, let's measure progesterone in the saliva. Turns out that the assay is not validated. So you don't want to go where there is not a validated assay before doing research of that, proposing a research, because somewhere somebody in the study section, for example, David Wright reviews my grant, he'll say, Jaideep, don't you know that assay doesn't really work? Because he's done progesterone research before. So that's the kind of thing you have to make sure that it is feasible to do before you go in. Similarly, technology analytics, if you're going to use some complex analytic technique, make sure that it's been used and it is feasible to apply it. And any innovations that are not published definitely need to go in the preliminary results section. Second sort of approach thing is, how will you accomplish your aim? So make sure that your inclusion criteria, exclusion criteria, enrollment mechanism, and intervention are very clearly laid out. So when you lay out your inclusion criteria, make sure that they are congruent with your research hypothesis. So you're including the subjects who are likely to be appropriate for the study question that you asked. Every exclusion criterion that you lay out, give a rationale for why are you excluding those subjects, and what is the importance of excluding them. Enrollment mechanism obviously is important. If you have robust enrollment mechanisms, you're likely to enroll sufficient subjects into your study. If your reviewer believes that you don't have mechanisms in place to enroll subjects, they're likely to be doubtful that you can successfully complete your trial. Clearly lay out what intervention would occur and in what sequence and how. So in laying out your approach, in writing the grant, make sure that it flows from inclusion, exclusion, enrollment to intervention smoothly, and all those things are clear to the reader as to how the study would flow. A good approach is to lay out a diagram or a figure illustrating how those steps will proceed from enrollment of the subject. It may begin with screening, enrollment, inclusion, exclusion, intervention, and then go on to primary outcome determination. If you can lay that out in a figure, it's much easier for the reviewer to read as to how the study would flow. Primary outcome is critical. This is what is going to determine the success or failure of your study. So it should be very clearly stated. And I can tell you, in discussions regarding the primary outcome should take several hours for you to do, ideally with some experienced researchers and somebody who's outside the field who can say, oh yeah, that sounds reasonable, I think you can do it. There should be clear evidence that that outcome is feasible, it's reproducible, and it is sensitive to what you're trying to measure, and it is also specific. And these criteria speak to your rigor as a scientist. Rigor and reproducibility have become an area that NIH is very concerned about. They require all reviewers to assess every grant for rigor and reproducibility, and that's where you get to show that you've thought about your study design and your primary outcome very carefully, and it will be something that can be done. There can be a series of secondary outcomes. As you all know, your secondary outcomes can be such that your study is not powered to determine a difference in the secondary outcomes. On the other hand, they add to the study and provide supplementary data that would be helpful. So, but even in providing secondary outcomes, make sure you justify them. Remember that in doing the analysis for secondary outcome, there will be a certain cost associated. So in your mind, do a cost-benefit analysis as to why you want that secondary outcome to be done. Statistical analysis plan is often in a separate application. However, make sure that in the primary application as a clinician, you justify the expected effect size and sample size. And it's very apparent to reviewers if you're going to do if you inflate your effect size to deflate your sample size, or when you've not done a sample size analysis, very commonly you get a sample size of 100 or 200 or 300, some very even number, which is a sample of convenience rather than expected effect size worked out rigorously. That shows lack of rigor. Again, recruitment, I spoke about it earlier. It is a common problem. Make sure that you support your claim that you can recruit sufficient number of subjects. Show that you are realistic. A common error is to be overly optimistic. A common error is, we have 300 such patients in my hospital, I'll enroll all of them in my study. You'll probably enroll 30 out of those 300 if you're lucky. So might as well be honest and go with that. Briefly address inclusion of women, children, and minorities, and the safety plan. A longer description of these follows in human subjects, but in the main application, it's good to address that those issues have been taken care of properly. And if you're going to exclude any of them, explain why very clearly, and again, justify their inclusion or exclusion. The environment is your opportunity to demonstrate that adequate institutional and network resource support exists to do the research. Again, what kind of clinical resources exist at your institution? Do you have a CTSA? Do you have a NeuroNext trial? Do you have the stroke net, as David talked about? Do you have Siren as a hub? All those are institutional resources in addition to things like clinical trials offices, the old units for doing inpatient clinical research, whether all those facilities that exist. And often these resources, a list of these resources are available in clinical trials offices, and you can go ask them and they can provide you with these. If you're going through a network, then you have to list your network resources and individual site resources. Again, this is your opportunity to show that infrastructure exists to support the research you're proposing. Investigator is another scored criterion. This is your opportunity to tell your story. Within your BioSketch, it's no longer very new. BioSketch allows you to list your strengths and weaknesses. If there are any weaknesses and there are parts of the trial or the study that you're not an expert on, make sure that you address it directly, head on, and ideally what you do is you use collaborators who can help with that area of expertise. They should be either co-PIs, joint PIs, or write a collaborator letter to provide that they will help you out with specific aspects of the grant. And this is important to make sure that you have an appropriate group of people to approach this research. And of course, BioSketch is your opportunity to list areas of expertise. You can generate your BioSketch using an online tool, MyNCBIs, if you wish. In preparing your grant, make sure that it is timed appropriately so that you go to grant officials, people who help you prepare the grant, prepare the budget. If you have more than one site, they have to negotiate a site budget, so get support letters, get subcontract negotiations going. So bigger the grant, farther out you need to be in planning for it. You should prepare your budget early and revise it if you need any new items as you change it. So if you've prepared the budget early on, you can at least amend as you think of new things. If you're going to have multiple sites, make sure that you ask for your budgets from other sites early in the process. It's very difficult for other sites to do things very, very quickly. I know institutions where it takes a month to get something done, and that's not uncommon. Screaming at people doesn't make them work any faster. They typically tend to slow down. So you have to justify every item in the budget, especially if the budget is non-modular. If the budget is more than $500,000 per year, you have to get NIH permission and make sure you meet with institutional offices to get support letters as you need them. So once you've submitted the grant, it goes for review, and David will go over how the review process works, and then you will get a summary statement back, which is summarized right here, which will have a summary of the discussion about how your application was discussed. This is what was said about it. Then the written reports from 3 reviewers or more, which will have scored criteria and scores for individual criteria, including significance, investigators, innovation, approach, and environment. After the scores are given, they discuss all the other things like human subjects, all the regulatory concerns, budget, and period of support. Even though budget is discussed after scores, always remember that reviewers have budget in mind when they score a grant. When a grant is asking for $20 million, they would be very tough than when it is asking for $2,000 or $200,000. That's psychology. That's even though we are repeatedly told that this budget will be discussed after period of support, but people have read the budget when they're scoring a grant. So I've included some online resources. One is from NIAID, where they've put some sample grants on their website, and they've even given some, these are actual funded grants, so they've given summary statements. You can go read some of these. And then Nature published an article on grantsmanship. Why I like this one was it talks about clinical grants. Most of the grant writing guides, etc., focus quite a bit on bench grants, and there is a different art to writing clinical grants. So I'll stop there, and David can start. If you have any questions, I can answer now or later.
All right, thank you, Jaideep. That was awesome. I think what we'll do is we'll go through my part, and then we will share my screen here and then get the slides up. All right, Jaideep, can you see the slides? They look right?
Not yet. I can just see you.
Just see my face? Okay, let's try it this way. Share screen.
Now we can.
All right. Now let's see if you can see now. How's that?
Yes, it works.
Okay, very good. All right. So when we were asked to do this talk, there were a number of things that we were asked to talk about, and these are the sort of 4 things that I'm going to just discuss. So what study sections love and hate? What happens during the study section? What are the review criteria, as Jaideep already sort of went over, so we'll just sort of briefly go through those. And then what is the dreaded ND? So let us move forward. So what does study sections love and hate? And I think Jaideep really touched on most of these. I think I just wanted to kind of highlight a few. Probably the dagger in a grant is if it's sloppy. I can tell you that a lot of folks are trying to read these in between their clinical practices and writing their own grants and all the other things that they're trying to do. And when they pick up a grant that doesn't read well, it's an instant negative, and it doesn't matter how good the data is inside of it. If you make the reviewer work harder, you're going to lose. That's the bottom line. People do this as volunteer. They actually don't really get paid for it other than some honorary kind of money that goes to travel, which is less than actual the cost of the travel, to be honest with you. But you need to make sure that you tell the story, you tell it very convincing, and it's beautifully written. So I would encourage you to get outside folks who don't know anything about your topic and have them read it. If they can understand it and it makes sense to them what you're trying to do, then you've got a winner. If everybody is confused or they don't understand a section, don't try to justify it, try to fix it, and pay attention to that. Another one is that it's too dense, that you sort of are pushing the limits of what is allowed, like font sizes going all the way out to the edges of the paper, not having any spaces, no graphical images or anything like that. It may be a great grant, and actually I've seen some fantastic grants like this that actually got really good scores, but it makes it really hard on the reviewers. And remember, part of your job is to make it easier on the reviewers. You want them to like your grant, right? So when they read the grant, in addition to its content, you want to make it something easier for them to read. Jaideep mentioned overly ambitious, especially for junior or training grants. If you've got more than 3 aims, there better be some really good reasons for that, and they make absolute sense. Otherwise, you're going to get this overly ambitious comment. Dependent aims is another dagger. In other words, if you have aim 1 that you have to do before aim 2, it makes sense. Or if aim 1 fails, aim 2 is not important to do anymore. That's a no-no, and that will instantly kill your grant. So somehow you have to make them as parallel aims if you're going to have more than one, so that no matter what happens to aim 1, aim 2 still makes sense. Lack of serious statistical support. I know a lot of you guys out there have MPHs and PhDs and other things on your name tag, but I'm going to tell you that the statistical reviewers in the room want to see a statistical reviewer on the grant. I don't care, really, honestly, how good the statistical section is written. If there's not a statistical person on your grant that's getting more than 3 to 5% effort, the stats guys are going to kill you. They're going to go, well, this doesn't have a serious stats team, we're not going to do this. So that's just a little warning. Make sure you involve your statisticians early, and make sure they actually are a part of the planning of it, not just at the end when you ask them for a sample size. Poor justification of your approach. I won't go into this. Jaideep covered this extensively, very well. And he also mentioned outrageous budgets. As he mentioned, these are not supposed to go into your scoring, but since the reviewers see your budgets as part of the proposal before scoring happens, that bias is there. So just make sure that it makes sense. Don't pad it too much. That will be very obvious to experienced reviewers. And then if you don't put in information about human subjects, now within grant review, you actually have to describe how, if you don't have enough diversity in your recruitment, how you're going to go about and get more diversity. You can't just simply say that your plan can't be, we're going to seek more diverse patients. You actually have to have a plan in there that if you, unless it's part of your study to look at one specific ethnicity or gender, you've got to have a plan to figure out and justify how you're going to go about finding more of those patients. So what study sections love? Well, kind of the opposite of what they hate, right? So they like it clean. They like a very well-supported story. So unless you're frankly, if you're an R21, which is supposed to be exploratory, they're still going to look for some preliminary data or some evidence that you can actually accomplish your study or your goals. You want to link things for a study section. You want to make sure you go through each section. There needs to be no dotted lines here. You need an absolute connection from the significance to the foundation to your approach to your outcomes and how you're going to interpret your results. And then some thoughtful discussion on limitations. So if there's a limitation in your study or in your plan, they will find it. If you mention it, you'll go a long way in sort of mitigating their concern. If you don't mention it, they're going to say you're naive and don't understand. So just be aware of that. As Jaideep mentioned, they want to make sure that you can do it. So as he mentioned, the 10% rule. So if you have 300 patients, the sample that he used, that you think could be recruited based on doing your preliminary study at your site or wherever your sites are, take 10% of that, and that's really what you can enroll. People try to take half, they try to take 60%, and I'm going to guarantee you that's going to get you next. And the truth is that you're going to be closer to 10% than you are half. So just be aware of that. And then if you're doing a go, no-go grant, in other words, if this is a phase 2 trial where the question is if positive or if you get what results will make you go towards a phase 3 or a second phase 2, but mostly it's a phase 2-3 study. If they say they want a go, no-go statement in there, you better have a go, no-go statement that's very clear and easy to surmise or develop from your results. If I get X% positive result, we're going to go forward. So that needs to be in there. I'll just move on. Okay, so what happens in study section? For most folks, unless you've actually been in study section as a reviewer, it's a black box, and I get it. And it's kind of scary sometimes when you get back the reviews and you go, oh my God, what were they thinking? Usually it's really, I just want to try to demystify a little bit. Many people sort of think about it as this room full of these guys who are trying to do everything to kill your grant. In part, that's probably true. As Jaideep said, the reviewers really are looking for problems in your grant, which I hate that aspect of it. I wish it was more, what can we find that's positive about a grant and then move forward. But the bottom line is they're looking for holes in your grant. So if there are holes to be found, they will find them. They may even find some holes that you didn't think there were there or that you disagree with, but nonetheless, you don't have a say in this. It's the reviewers. So it's sort of like the old movie 12 Angry Men. I'm sort of telling my age. Actually, this is way older than I am. But in any case, this is a review panel. This is what it really looks like, except nowadays there's less paper. They've gone to all digital now. Used to, every single grant was printed out and people would bring them in stacks, and you see the boxes in the center where they would throw them into after review. But there's usually a very diverse group of people in the room. You will be assigned several reviewers specifically to your grant. So you will have a primary reviewer, and then you will have, depending on the size of your grant, a number of secondary reviewers. That can be anywhere from 3, 4, or 5. Everyone else that you see in this room, though, will vote on your grant. How does that work? Well, your primary reviewer will give the story. It is essential that your primary reviewer believes in your grant, because that is the individual that will essentially fight for your grant. And if they don't, then the chances of you going forward are really almost zero. The secondary reviewers are on the panels. They have a chance to sort of argue that your grant is good or not good. Everyone else in the room, although they have an opportunity to read your grant but weren't assigned to it, and not likely will read your grant, they'll be reviewing a little bit as the proceedings are going on. They listen to the story of the people who actually review it. and then they will make their scores based on how convincing that is. What do you mean by scores? So here is what the scoring is. High impact, score of number 1 is a perfect grant. There are a few of those actually. This is exceptional with 0 errors or 0 problems. And then a 2, which is outstanding, 3, excellent, as you see going on. And the median gets around 4, 5, and 6. A 9 is essentially don't come back, but not technically. Here's how these actually track, though. So when you look at this, this doesn't look too bad, all right? So strength, weaknesses. But here's the issue. So these actually track to the number of weaknesses in your grant. So literally, if you look at the weaknesses that were found by the review panel, they sort of add up these weaknesses and they consider them or codify them as negligible weaknesses, minor weaknesses, moderate weaknesses, and major weaknesses. No weakness is an exceptional grant. Minor weakness gets you into the outstanding excellent range, and that's only 1 or 2. If you have more than 1 or 2, then you're already at a 4. That's minor weakness 1 or 2. I mean, more than one or 2. So you see that it doesn't take very many weaknesses to get you out of a fundable range, because technically 1, 2, maybe 2.5 is in the fundable range. Once you get to the 4, 5, and 6, it's not fundable. Okay, so you can have a really good grant, very good. You're not fundable if you have numerous minor weaknesses. If you have one single moderate weakness, you're already up into the 5s. So that's why it's so important to be very careful with your grant writing, because this is literally what the review committees are being held to. So what happens in study section, and this is just sort of brief, almost all study sections operate this way, although there are some inherent differences. So first thing that'll happen, comes up to your grant, everyone who was the primary and secondary reviewers will give their initial scores that they reviewed when before they came to the review section after they read your grant. So initial scores will be called out. Then primary reviewer number 1, this is your person who's going to advocate for your grant, gives an overview of the whole grant. So what's the significance? What's your background of the grant? Why are you doing it? What are the outcomes? What was your approach? And then lists sort of what the weaknesses and strengths of the proposal were. So that's the primary reviewer. Usually then, now at least in clinical trials, there's a secondary, a scientific foundation reviewer, that if you are going towards a phase 2 or phase 3 trial, scientific foundation reviewer gives the strength of the evidence from preclinical to move forward. In other words, how many animal studies, how strong were the animal studies that were done? Has any other translational studies been done? Has any smaller pilot human trials been done? What is the strength of that evidence? And so that scientific reviewer who hasn't read your isn't really giving you a score. They're just basically trying to provide evidence as to how strong it is to move forward. And then, this came about actually over the past few years because of the failure of going from preclinical to clinical of so many trials, especially in the neuro space, that they said, hey, why don't we have a basic scientist come in here and tell us what's the strength of the evidence to try to prevent so many failures. I'm not sure that has really worked as hoped, but nonetheless, these folks still present. Then your assigned reviewers, 2 through however many you had, will give their additional comments over and above what the primary reviewer did. And sometimes this can go on and on and on. But for the most part, they're supposed to only give anything additional or maybe even some arguments that counter what reviewer 1 gave. And then the floor opens to the rest of the group. And so people who didn't review your grant primarily or secondarily will ask questions to the reviewers who did and try to clarify some of the problems or issues that they heard come up And sometimes they hold the group accountable. In other words, if there were multiple minor or even a couple of moderate issues and the folks originally scored it pretty well, the other group is going to say, wait a minute, that doesn't sound like a good grant. They need to fix a lot of things. And so they'll actually argue for an adjustment of the score. And then the final points are assigned by the people who actually did the original reviews. And so, in other words, all of this discussion can lead to changes in your scores, and often does lead to changes in your scores. And then everybody votes and puts in their scores. This is done. They score it without anybody knowing what the scores are. So everybody in the room, even the ones in addition to your primary and secondary reviewers, actually enter a score into the system that goes to the program officers. And then after that's done, they close voting, and then they discuss budget, human subjects, and other issues. So that's sort of the process. Each grant mechanism may have some specifics, different issues that they're addressing, like if you're in an STTR or SBIR, which is small business loans, or small business grants rather, they may be talking more about intellectual property or transition plan or other things. So you can see those, and you just go on the website. This I pulled off of the NIH website, but you can see some of the different categories. So as Jaideep mentioned, here are the individual sections that you get scores on. So you get an overall score, which is really not an average of the individual components. It's more of, and this is the question that the reviewers are asked to ask of themselves, which is, how likely is this grant to be successful, and how likely, if it is successful, will it change practice? And so that is sort of the overall gestalt, which can be pretty different than the average of the scores they gave you. There are individual scores for the significance, again, how much will it change practice, and so on. The approach is really where all the money is. So, investigators, innovation, environment, they get a little bit of playtime, but mostly everything is spent on the approach. And that's where most of the review is. And so what is ND? So not discussed is ND, and often you will get back your grant. In fact, at least half of the grants that go into the review session will get an ND. And typically this is because there's not a lot of time to review all the grants in front of everybody. Now, you've been assigned a primary and secondary reviewer. They've done a review and they've written a full review. So if it's not discussed, you get that review. What you don't get, though, is the other 20 people in the room and their comments or a vote for a score. So basically, the not discussed comes from the scores that you were originally given by your original reviewers. So what is that? That's usually a score of 5 or greater. So if you didn't make it 1 through 4 on average in your scores between your reviewers, you're probably going to get an ND. Remember, an ND does not mean that the grant is dead in the water. At least it is probably for funding that go around, but it just means you have several minor or a couple of moderate weaknesses that you need to address. So it doesn't mean that you shouldn't resubmit. Just read the reviews very, very carefully. I create a grid in Excel where I have every sentence broken down by every complaint within every sentence, and then how I'm addressing that issue, and then where the change is in the grant that I resubmit, just to make sure that I'm thorough and that I didn't miss anything. It is okay. You can reapply. In pretty much all of these, unless they tell you in there, which would be really a big hit, not to reapply, which is pretty rare. I show this not because I'm David Wright, this is David Wright, but because grant writing is baseball. And I don't know how many out there like baseball, but essentially a batting average of 2 to 3 out of 10 is a great batting average. That's what the pros have. So just FYI, don't give up on your first grant, your second grant, or your third grant. Remember, it's 2 to 3 out of 10, and you get better as you write grants. So really when you start getting into your 7th, 8th, and 9th is when you should be hitting. Maybe not necessarily your 1, 2, and 3. Now, I don't want to scare people, but that's the truth of it. And just don't be frustrated by not getting it the first 2 or 3 go rounds. And that's pretty much it that I have. And I guess we can open the floor to questions if there are questions. Lisa, are you driving this part?
Yes, Dr. Wright. One of our participants is asking, does the scientific foundation reviewer have access to the grant, and do they review it? Based on the patient population of interest, some preclinical data may apply and some may not. For example, I am studying on only patients with successful endovascular stroke treatment. Thus, only rodent models of transient LVO, but not permanent LVO, apply, as physiology and hemodynamics in these two models are very different. Thanks.
Yeah, so yes, the scientific reviewers do review the grant. Typically, they are experts in the field, so they will know these differences in the models. I mean, that's why they're picked and chosen, because they're experts in these particular fields. Yes, the answer to that is yes, they read it.
We have another question. Thanks very much. Could you please describe the review process for resubmitted applications?
It is exactly the same. You get assigned a cadre of reviewers. Hopefully, you're assigned the same reviewers. That's your best chance. Doesn't always happen because the same reviewers aren't necessarily there going or attending that next review cycle, which is several times a year. But if possible, the program officer will try to reassign your grant to the same people who reviewed it. And I'll tell you why that's better. It's better because if you address the issues, typically the people who reviewed it before will give you a pass if they like your readdress. The problem with getting new reviewers is they may find new problems that weren't addressed, which are really annoying, actually, even as a person who's in review, when someone starts bringing up new problems in a grant that weren't picked up before. And to me, it's almost not fair, but happens. So yes, it's reviewed exactly the same way.
Okay, we have another question. Could you clarify the 10% you mentioned about sample size based on your previous sampling history?
Sure. So that's the rule of thumb that I use. If let's say that I'm doing a stroke study, for example, and I have certain exclusion and inclusion criteria, and I go and I estimate how many patients come into my hospital with at least those inclusion criteria and some of the major exclusion criteria that I'm going to have, and I count that number. Whatever that number is, I take 10% of them is how many I'm going to be able to enroll. That is the most conservative, but frankly, the best rule of thumb that you can use. Jaideep, do you have any comments on that?
No, but I think it's better to be realistic than you won't get challenged on that. Is my audio on?
Yeah, you're on. We hear you. Yeah, I think it sounds really, really conservative. But the truth is that you're closer to 10% than you are 50%. Because first of all, you got to start considering consenting. You got to find all of these patients, because most of the time when you looked at your data, you did a retrospective review, and so you found them all in retrospect, but you're not going to find them all in prospective. And then there are going to be some exclusion criteria that you just weren't able to measure very well that are going to knock out even more patients. So when you add all those things up, it doesn't add up to half the patients. It adds up to less than half. And so it's just a better way to do it.
Okay, we have another couple questions here. Does the personal statement in BioSketch have to be specific to the research project, and does it have to include details on mentorship that I get, especially if there are mentors in other institutions?
I'll answer that as, and then I'll give it to Jaideep. Yes, it needs to be, the whole purpose of the new BioSketch method is that first paragraph is meant to be specific to that grant. It shouldn't be general like it used to be in the past. And if you are a new investigator, it is helpful to have some mentoring aspects in there, but you're going to have a different section for that. Jaideep, you want to?
Yeah, exactly. Make the personal statement very specific to the proposal you're going to propose, you're writing, and then the mentoring part should be in the mentoring plan anyway. In the K awards, there is a separate mentoring plan. So all you have to do is to mention your mentor or not, because their BioSketch will go. In fact, your mentor's BioSketch should be in there. And if the mentor is at a different institution, somewhere in your mentoring plan, you have to address how you will establish communication, because there's skepticism as you'll never see them or you'll never talk to them. So what is the mechanism for allowing you to be mentored remotely?
Any other questions, Lisa?
Yes, I have one more.
Okay.
Can you please comment on any specific details of review relevant to a standing clinical trial study section as compared to a CSR R01 committee? These applications tend to be quite complex, expensive, and the question of will this change clinical practice comes up often. Appreciate any thoughts for applicants.
Jaideep, you want to take that one?
I haven't been to that study section. You have, so go ahead.
Yeah, no, it's complicated. If you're talking about these multi-site study sections, it's or SEP as is another special emphasis panels, or another version of these. They are complicated, and the interesting thing that makes this hard on NIH is that in today's world, we do so many collaborative grants that many of the reviewers, it's hard to find reviewers. In fact, because in some of these big stroke grants, everybody who's doing stroke research, who should be doing stroke research, are a part of the grant. So it's it makes it really hard. And so if I'm getting your question correct, again, the bottom line is just write the best grant possible. And if you're doing it through some of these networks, the network individuals, certainly for Siren and for the Neurologic Emergency Treatment Trials Network of old, there's a lot of support from the network itself in developing and helping with your grant. They've been there, done that, so they know how to get successful grants through NINDS. If I didn't answer the question, can you ask it in a little bit different way? Or if there are any other questions, please, now's your opportunity. It's 12:54, so you have 6 minutes. Lisa, I see people are beginning to drop off.
Okay, so I appreciate your answer. Seems like the key threshold is being clear how a trial will actually impact clinical practice. Is that your experience?
Yes, absolutely.
Absolutely.
Yeah, if you can make an argument that you will change clinical practice or significantly improve. I mean, this isn't. I mean, very few grants, trust me, that get through NIH, of course, as you probably well know, really truly change clinical practice. There are some, You know, but the argument that you can make for not, you know, they're looking for not just incremental improvement, they're looking for significant improvement and change in the field or the lives of the patients that you're addressing. And if you can show that, then you will go a long way towards that overall the overall grade on your grant. Really, remember that I said it wasn't a mean of the individual scores, but the overall score. Really, a lot of that has to do with the impact that you can potentially make with a successful grant. The two components are how much would this move the field if it were positive? And then the second piece to that score is how capable or how likely is the trial to be successful. So those are really the two pieces that go into your overall score, even with these individual scores. Those are the big ones. So if you can move practice or change practice or lives a lot, then it's a high risk grant with a high reward, and that goes a little bit farther than if you really can't move practice a lot, or change practice a lot. Conversely, though, if there's a low likelihood that the grant will be successful, no matter how important, then you're going to get a negative score or a lower score. So just be aware of that.
Dr. Wright, we have another question.
Okay.
As a reviewer, what would you say is your favorite organization format for the research strategy?
Favorite format for the research strategy? I don't know that I have one. It's just a grant that makes sense. Jaideep is laughing. I actually like images. Not images as pretty pictures, but I like flow diagrams or things that help me walk through the thought of what the investigator has in approach section. I like to see a description of how they're linking their enrollment, where they're finding the patients, how they link their enrollment, how they're going to do the outcomes, are they validated outcomes, and what those outcomes are going to mean. Just very simple stepwise process is for someone who's not very smart like I am, it's nice to have a clean, clear, simple approach.
I mean, as David said, remember, if you want to help the reviewer, that primary reviewer is going to have to present your grant to the whole group. And if they can't understand it, they can't explain it. And if they can't explain it, then the rest of the group doesn't get it and it goes downhill. So if you make it so simple that your primary reviewer can take that grant, they have to literally be your best advocate at the study section and arm them with everything you have so that they can advocate for you.
Yeah, I don't think there's any one strategy that's better than another. What you put first, whether you put your inclusion exclusion criteria first and then walk through. I mean, to me, that's the way I typically write a grant is from the very point of entry of a patient into the system. And then I'll walk the reviewer all the way to that patient at the very end when their outcomes are being done, and then the analysis. Doesn't have to be that structure as long as it makes sense. And you may, I think there's one point, a lot of the basic science will do, they'll put the aims within each, or their hypotheses within each aim and divide it up into those different types of sections. Again, I've seen some beautiful grants written that way. For clinical trial, doesn't typically work out quite like that. Makes it a little bit harder, but.
It's 13 hours.
All right, it's 1 o'clock. Anything else before we sign off? Feel free to reach out to Jaideep. He's happy to answer all your emails on an ongoing basis.
Thank you very much, Drs. Wright and Kapur. We really appreciate it.